So yes, I'm going to talk to you today about IgA and how we think that a mucosal immune response, not only to SARS-CoV-2, but eliciting a mucosal immune response through vaccination is a sensible approach to dealing with this pandemic. And I hope to convince you of that today. Okay, so just to get everyone on the same page, because I know this is a diverse audience, I want to remind you that the immune response to SARS-CoV-2 involves a number of different aspects. And as we all know, the immune system is kind of like a Swiss Army knife. It's not just antibodies, it's not just T cells, but it's a coordinated interaction between multiple innate and adaptive immune cells. And we know that as soon as someone gets infected with SARS-CoV-2, there's very quickly an innate immune response is activated, and that this can then coordinate with the adaptive immune system to result in T cell activation and the generation of cytotoxic T cells that can kill infected cells, as well as helper cells that can help boost a humoral immune response. And B cells are also activated, and some of these B cells will go on to become plasmablasts and plasma cells that secrete high levels of antibodies of these different isotypes, and they have different functions in the immune system for elimination of virus. So in terms of the actual immune response to SARS-CoV-2, this pretty much plays out as our textbooks have taught us. In that we have a rapid immune response that peaks in terms of the adaptive immune response around 7 to 14 days following exposure to pathogen. And during this time, the pathogen should be cleared by the immune system. But following that peak, there is a partial decline, but there's a very robust memory immune response that's generated in response to the virus. And you may recall early on in the pandemic, there was all sorts of worries that the virus would not induce a durable immune response, and that appears to be not the case. Most people do make a durable immune response of varying quality, and vaccination likewise induces a durable immune response. And so there's been lots and lots of studies on this by many of Kelly's and my colleagues, and this is just coming from Dr. Miriam Pepper's study that I like to cite a lot because it's just so beautifully done. And all of these components of the immune system are not only activated but retained following SARS-CoV-2 infection. But really, we have a not as good an understanding of how this virus elicits immunity in the upper respiratory tract. And the upper respiratory tract is where we confront the virus, because we know that the virus is airborne and can enter our nose and our mouth. And indeed, if we think about where the virus infects host cells, this is through, we need the cells that are infected, it's express this ACE2 receptor as well as this other TMPRSS receptor. And these, the expression of these proteins is actually very elevated within the upper respiratory tract. And in particular, I'll draw your attention to the salivary glands, where you can see very high levels of ACE2. And so given the pattern of expression of these obligatory entry receptors, it's really important to have a handle on how the immune response plays out in the upper respiratory tract. So mucosal tissues have sort of a different flavor of immune response, whereas systemic immunity tends to be dominated by an IgG response. Mucosal immunity really invokes predominantly an IgA response. And this IgA response you can see at a number of different mucosal surfaces, not just the oral and nasopharyngeal cavities, but also the gut and the lungs. Whereas in the periphery, and here I'm talking about the spleen and the peripheral lymph nodes, you tend to get more of an IgG response skewing. So the reason why I like IgA, and I think IgA is just such a great defense against mucosal pathogens, is because it's actually got a different structure than IgG. So if in a normal, so if you look at IgA in the blood, it's going to be a monomer. And so that means there's 2 binding sites for antigen. But if you look in mucosal secretions, such as in the oral nasopharyngeal tract, there's actually 4 binding sites for antigen. And that's because IgA can form a dimer, and it's attached, 2 IgA molecules are attached to each other through this J chain. And as these dimers are detected by this polymeric Ig receptor, which is expressed in the submucosal tissue by epithelium, the IgA gets transported from this side of the epithelium through the epithelium and into the mucosa itself. And when it does this journey, it takes along a hitchhiker, it takes along the secretory component. And I'm going to come back to that because that's going to be a key piece in terms of some of the assays we've used to measure mucosal IgA. But when you see that there's secreted IgA, that is IgA dimers bound with a secretory component, that's a sign that that IgA is being made locally. And IgA in the mucosal surface has the potential to provide sterilizing immunity because it's really the firewall between where we're confronted with a virus and the systemic immune system. And I like to quote Akiko Iwasaki, who likes to say, it's like putting the guard outside of the door. I like to think of it myself as a bit of a bouncer, and it just keeps the pathogen from potentially entering into the body. So obviously it would be preferential if we can elicit this type of response in order to confer sterilizing immunity. So as I mentioned, we first started studying this at the beginning of the pandemic because we had different assays in place for looking at mucosal immunity. And we decided that we wanted to look at saliva as a biofluid. And saliva is very rich in secretory IgA. And we were curious as to whether antibodies to SARS-CoV-2 could be detected in the saliva. So Balata Isho, a graduate student in the lab, worked really hard at setting up these saliva-based ELISAs. They're a little bit tricky because saliva is, the titers of these antibodies are much lower in saliva compared to serum, so you need a very sensitive assay. And we also did some important pre-clearing steps to remove any background level binding of the saliva. So those were 2 really important modifications. So it wasn't really a standardized ELISA assay, but we got it to work. And what we would do is that in each plate that we performed the ELISA, we always had a pool of convalescent saliva as a positive control that we used throughout each assay. And then we performed a normalization, and that's how we reported all of our data. I'm happy to go into that in more detail during the question period if there are questions. But what we found essentially was that an IgG, an IgA, and an IgM response was observed in the saliva of SARS-CoV-2 convalescents. This is a mixture actually of acute and convalescent patients. And the IgG response was relatively stable, but the IgA response tended to drop off much more quickly than the IgG response. And very similar findings were found in the serum, and we compared our ELISA results with those of Nkyo Jingra, who is at Mount Sinai Hospital in Toronto, and they correlated reasonably well. And so that was very reassuring because we were using 2 completely different platforms, 2 different biofluids, but within a patient, we saw a very nice correlation between the saliva and the serum. Although I should mention that with the IgA, the correlation with the serum was not as tight as what we found with IgG. And that could be because the IgG that we detect in the saliva is likely blood derived, and it comes through what's called the gingival crevicular fluid in the mouth. And so when we're looking at IgG in the saliva, we're likely looking at a systemic immune response. But when we're looking at IgA, it's a little bit more difficult to know whether it's from the systemic response or from the local immune response. The other thing we've been doing is looking at the very, very earliest days post-infection. So this is work done by my student, Eric Cow, who's also co-supervised by Arthur Mortha. And we teamed up with Daryl Tan at Unity at St. Michael's Hospital, who was performing a trial where he was giving protease inhibitors, Kaletra, to subjects who had been contact traced. And if you remember earlier on in the pandemic, we had a shortage of swabs. And so he decided, well, I'll collect saliva. And then I found out about that. So I went and chased him and said, give us your saliva, because it's really hard to find people who are asymptomatic, who are infected, and you know they're infected. And so this was a really good way to look at the very earliest days post-infection, prior to the onset of symptoms. And what we found was in the people in red were the ones that were contact traced and became PCR positive, as opposed to the vast majority of people, which were in blue, who were contact traced but never became PCR positive. Here we've matched, but in reality, we collected about 120 different subjects at multiple time points, and only 12 of them became positive during the study, which I think is a testament of the public health measures that were being used at the time. And we were able to detect an anti-SARS-CoV-2 antibody response in the saliva as early as day 7, but the earliest response was really dominated both by IgM and IgA, whereas the IgG response slowly increased with time. And so this was most likely because this was an acute local mucosal immune response that was happening first. And so this again underscores the importance of understanding how this IgA response is generated, because it's likely to provide some protection at the site of infection. All right, so what do we know about vaccination? So I've shown you that antibodies are generated in saliva in response to SARS-CoV-2 infection, but very little is known about how vaccines, in particular intramuscular vaccines, which are really designed to activate the peripheral immune system, whether they would have the capacity to activate any kind of mucosal immunity at all. And our hypothesis was that no, they wouldn't. And I'll show you some data that in fact they do. So anyways, what we know about vaccines, and of course this is a very intensive area of research, is that, and we're just going to focus here today on mRNA vaccination, the antibodies that are induced by mRNA vaccine peak very quickly and very high, and then they decline. I refuse to call this waning, because the reason I don't like calling it waning is because in the background you have a gradual accumulation of memory B cells over time, and they actually don't decline, they still go up, especially as the immune system is boosted with subsequent doses. And that memory B cell response actually gains breadth over time. And so it seems that 3 administrations of an mRNA vaccine confer this sort of sweet spot in their ability to confer broad memory B cell repertoires that are flexible enough to even recognize variants as distinct as Omicron. And for that reason, 3 doses of vaccine seems to be effective at least preventing Omicron-induced disease. Infection is another question, and we'll get to that. Okay, so all of the right things are happening with vaccination. We're getting memory B cells, we're getting memory T cells, we're getting a nice antibody response that declines. We are getting a plasma cell response, and Ali El-Abadi has shown that nicely, that you can look and see plasma cells in the bone marrow of vaccinated subjects. But again, we don't know if this will actually induce any mucosal immunity, because that's really what you need for sterilizing immunity. The mucosal immunity here being whatever's generated locally and whatever's coming in from the blood through the gingival crevicular fluid. So we looked for that, and this is work done by Salma Sheikh Mohammed and Gary Chow in the lab. And Gary Chow is a project manager, and Salma is a graduate student who's about to do her qualifying exams in a few weeks. And we teamed up with Sharon Strauss at Unity, who received CITF funding to examine the vaccine response in long-term care staff and residents. And we focused on the long-term care staff because they were relatively age homogenous. And what we found, and these are folks that received their first and second doses at the regular monograph interval. So they did not have delayed second doses. They received their first and second doses either 3 or 4 weeks apart, depending on whether they got Moderna or Pfizer. And when we looked at IgG levels in the saliva and compared with our pre-pandemic controls or the COVID-19 convalescent and acute subjects, we saw a very comparable level of IgG in the saliva after 2 doses of mRNA. And as I said, this is probably coming from the periphery, from the blood, through the gingival crevicular fluid. But what surprised us was that we also saw a modest IgA response in the saliva that was not as high as what we saw in COVID convalescents, but was nevertheless detectable in about 30% of the subjects that we tested. And this IgA response was accompanied by secretory component. And that suggested that the IgA was not necessarily coming from the periphery, but could be generated locally, which we found really interesting, because if that's the case, that would imply that there's been a local immune response provoked by a parenteral intramuscular administration of an mRNA vaccine. But what was very curious is that now this is a different cohort I'm showing you here. This is what we call our MSB cohort, and it's enthusiastic local volunteers in the form of students and postdocs and PIs. Here we're looking at folks who had a longer interval between dose 1 and dose 2, like most of us had. So 3 months between dose 1 and dose 2 of the mRNA vaccines. And that gave us the opportunity to look at the immune response after 1 dose, because we weren't fast enough to be able to do that in the long-term care folks. And when we did, what we found was that 2 to 4 weeks post-dose 1, almost everybody made this IgA response. And it was associated with secretory component. So even though we only saw about 30% of folks making this response in the LTC cohort after 2 doses, if we back up and look after a single dose, you actually see that most people, if not all, make a secretory component associated IgA response to the mRNA vaccine. But the kinetics are so different than IgG. So this response peaks after 1 dose, and then it declines. And at 2 to 4 weeks post-dose 2, we don't see a boosting of this response. What we do see is that, again, about 30% of our cohort retain a secretory IgA response that's higher than background, but lower than what you would get with a COVID-19 infection. In contrast, if you look at IgG, the immune response is exactly what you would expect it to. It declines after 1 dose and then is boosted after the 2nd dose. So very different kinetics of IgA versus IgG responses in the saliva. So why would we see IgA, and specifically secretory IgA, in the saliva of a COVID-19 vaccinated person? So it's possible that these people have had COVID-19 and that you are eliciting some kind of memory recall response due to the presence of a tissue resident memory B cell. We don't think that's true, because we actually looked at the nucleocapsid antibodies in these people who were vaccinated. And some of them were nucleocapsid positive, and suggesting they had had a prior infection. And the metadata for some of these people was confirmatory of that signal. However, those who did not have an NP signal and did not have metadata to suggest that they had had a previous infection also had evidence of a secretory IgA response. And that when you looked at the difference, there was no difference between these 2 groups. And so we didn't think that that was the explanation for why we were seeing a local secretory IgA response. We also sent these samples to Samira Mubareka at Sunnybrook Hospital and asked, well, maybe there's an ongoing infection at the time of sampling, and that was not true either. And so we don't have an answer to this question, and Salma is now going to look at this question in the context of mRNA vaccination in animals, in mice, and using some nice antigen tracking systems, looking at whether antigen is distributed more broadly than we thought. That could potentially explain why this response is not boostable, because presumably after dose 1, you have very high levels of IgG that would opsonize a subsequent antigen boost. And so that could be why you saw this response, but it wasn't boostable, because maybe the antigen doesn't get there at dose 2 and beyond, because of this high level of IgG that's already in circulation. So that's one possibility. The other possibility is one that has been explored by in the older literature when people used to study yellow fever vaccine responses because we didn't have good ways of studying immune responses unrolling on our doorstep like we do these days. And there is evidence that mucosal homing receptors can be upregulated on immune cells that have been primed using systemic vaccination. So it could be something happening within the axillary draining lymph node that's programming some cells to go to mucosal surfaces. So those are the two possibilities we can think of, and I'm happy to entertain other possibilities. Okay, so why do some people have high IgAs? We saw this sort of proportion of people, 30%, that had this retained IgA. And we thought about kids and we thought about mucosal exposures. And when we think about a burden of mucosal exposure, anybody who has interacted with children or parents of young children will tell you that they're just living in a field of snot. It's just like one infection after another. So we were very curious about what this immune response would look like in children. And so when it was clear that, and Manish was great at guiding us on this, when it was clear that the vaccine was going to be approved in 5 to 12-year-olds, we quickly did a Twitter drive and were able to assemble a very eager cohort of parents and children who wanted to have their immune response measured. And so I'll just show you some of that data here. So we collected saliva from different time points from about 100 kids. And we looked at what this immune response looks like after dose 1, and we compared this to the adult cohort that we had collected locally. And it's not terribly surprising that the antibody levels after dose 1 are much higher in children than they are in adults for both IgG and IgA and secretory IgA, which I'm not showing here. So significantly elevated salivary IgG and IgA responses in singly dosed children. Now, we're still waiting on the nucleocapsid data from this cohort, so that's going to be important, but one of the entry criteria for our studies was no prior exposure. But we'll soon have the serum analyzed from these kids to really confirm which of these were COVID naive. So that was of interest. But when we look at the pattern of the response, the kinetic pattern, it's the same as what we see in adults. So here's the baseline, here's dose 1, and here's dose 2 for IgG. So you see a boosting like we saw in adults. But for IgA, here's the baseline. There's actually quite a lot of variability in the baseline for kids, which we think is quite interesting, and we're going to study that further. And then here's dose 1, and here's dose 2. So we have that crashing of the IgA response after dose 1 that we saw in adults, and now we're seeing it in a completely different demographic. Basically, the pediatric immune response, the take home here is that it's stronger, higher amplitude after dose 1, but it follows the same pattern as what you would see in an adult. So it's not like the fundamental rules of change, whatever those rules may be. Okay, so just to summarize this part of my talk, and then I have a few more slides. Are anti-SARS-CoV-2 antibodies detected in the saliva post-COVID-19 vaccines? Yes, not as high for IgA as what you would get with a COVID convalescent, but similar for IgG. Can they be mucosally derived? Yes, and that doesn't appear to require a prior infection. And does this happen in kids? Yes, but higher amplitude. So, you know, we've been really obsessed about this IgA response. And of course, one of the big questions is whether it even matters. Is it just, you know, something that comes along for the ride? Or does it actually have some protective effect at preventing infection? And that's really, you know, that's not what our vaccines were designed to do. We keep telling the media this. Our vaccines were designed to protect us against disease. And any protection against infection is likely due to high levels of antibodies that do decline with time. So really to understand what aspects of the immune response to vaccine are correlated with protection against infection requires prospective cohorts. These where you can catch breakthroughs, this is in the pre-Omicron era, Where you've been able to catch breakthroughs, these are tough. And believe me, we scoured the world. And so I'm going to show you some data on a couple of breakthrough cohorts and what relationship this has with serum antibody titers. Unfortunately, we didn't have saliva from these prospective cohorts. Okay, so the first cohort was assembled by Alison McGeer. This was part of Sharon Strauss's larger CITF-funded study. What happened was that there was a long-term care facility in Toronto where they had a gamma outbreak. Remember gamma? And in the outbreak, they were able to really segregate those who were exposed, infected, and exposed, uninfected. And so this was the same virus in the same living situation. And from that group of exposed individuals, there were 5 breakthroughs, and there were 12 that did not have the breakthrough, but were nevertheless exposed. And then cohort 2 is a case control cohort out of Sheba, Israel, where subjects were vaccinated, and then people were monitored throughout a monitoring period for breakthroughs that were PCR confirmed. And we received 11 breakthroughs and 56 controls, so a bigger cohort. The important thing we're looking at here is what level of antibody was induced by vaccination at 2 weeks post-dose 2 prior to that breakthrough. And is there any correlation between the level of those antibodies and subsequent breakthrough status? And I'll mention that at that time point, 2 to 4 weeks post-dose 2, the saliva and the serum antibody responses correlate with each other. IgG correlates extremely well. IgA positively correlates, but less well, but still significant. And these are the folks that helped with this. I mentioned Alison McGeer and then Gili Regev and her perspective case control cohort in Israel. And the serum results were analyzed by Andrew Gingras and Kento Abe, both at Mount Sinai. So here's the long-term care cohort. Looking here on the left is the IgG antibody response to the vaccine in those who were exposed uninfected versus exposed infected. And there's no significant difference in IgG levels either to spike or the receptor binding domain antigens. When we look at the IgA response, we started to see a hint that there was a reduction in IgA in those that were exposed infected versus exposed uninfected. But it did not hold up to a multiple comparisons test. And so it wasn't statistically significant, hence the look for another cohort. Here's the Sheba cohort, which is more powered. We see the same exact thing. So this cohort did not show any predictive value for IgG at 2 to 4 weeks post-dose 2 in terms of whether you would be infected or not. These were mostly alpha breakthroughs and some delta. Whereas if we looked at IgA, now we see a statistically significant reduction in vaccine-induced IgA responses in those people who subsequently had a breakthrough infection. And this is a summary of those 2 cohorts. And what was really gratifying to me was that it's basically these data could be photocopied. You just add more dots, and that's what you get with the Sheba data compared to the Toronto data. And yet, the Toronto folks were aged, the Sheba folks were not. The Toronto folks got Moderna, hence the higher amplitude of the antibodies. The Sheba folks got Pfizer. And then different geographical regions. So in spite of these differences, we saw the same pattern, with that pattern achieving statistical significance in the more well-powered group. So yes, I think IgA matters because we don't have great correlates for protection against infection. We have decent correlates for protection against disease, and neutralizing antibodies being really one of the things we can hang our hat on. But in terms of protection against infection, we're groping a little in the dark. And I think IgA is really going to be important based on these data. So how are we going to do this? Well, as I said, only about 30% of vaccinees retain an IgA response in the saliva. And since that is where we are exposed to the virus, that's, if IgA is important, that's where you would want it. And to do that, we really would need to deliver, to do that reproducibly and faithfully, we would really need to deliver a vaccine through an intramucosal route, intranasal route. And there are some vaccines, this is likely out of date, but this is taken from the New York Times vaccine tracker. They did not include the McNagny vaccine, where they are boosting with an adenovirus. But basically, there's actually not a lot being developed in this space. And I think Canada should really get in there personally. And so I think that really the secret, however, is really a prime boost strategy by starting systemically and then going mucosally. And that's borne out by a couple of papers, as well as this really nice preprint from Akiko Iwasaki's group, where she showed that if you prime intramuscular and then come in intranasal so if you prime with mRNA and then come in intranasal with a mucosal boost, you can really build on that systemic immunity that was established through the prime to teach the immune system to go to the mucosal surface. And that's where you could potentially provide protection against infection. And that proved to be true in her animal experiments. So if she did it that way, and she boosted with either mRNA or even unadjuvanted spike trimer, she was able to induce a local immune response in the nasal tract. And she was able to protect these mice from infection, even infections with viruses that were very different. So for example, SARS-CoV-2 variants, but even SARS-1 was able to, this prime boost strategy was even able to protect against SARS-1. And so as we start thinking about pan-sarbecovirus approaches, I think we have to be thinking about the mucosa, because that might provide the kind of flexibility at the mucosal surface that you would need, provided by this IgA dimer with this long hinge region that's nestled in a mucosa and provides that potential sterilizing immunity. So we wanted to, this is just my last couple data slides. So we wanted to ask, all right, can we do a sort of proof of concept that this could be happening in humans? And we thought, well, why don't we compare people, and there's now lots of them, who've had 2 doses of vaccine and then had an Omicron breakthrough. And we all know that these were happening around December, January, as compared to people who had 3 doses of vaccine with their dose 3 happening around the same time. And I know I had my dose 3 in December, and that was when everybody was getting Omicron. Well, not everybody, a lot of people were getting Omicron breakthroughs. So this is kind of a nice comparison group. And we're right now in pillar 1 of COVARNet. We are, looking at these subjects that I'm showing you here and just taking them all apart and looking at the T cells and T cell response, et cetera. So I'm just going to show you the saliva antibody response. And what we found was that, not surprisingly, if you look at an Omicron version of spike protein, a vaccinated person who's had an Omicron breakthrough makes a better Omicron spike response, whether that be IgG or IgA. And that's not surprising because Omicron is so different. So it's teaching the immune system to recognize Omicron spike, and that would work better in someone who's had the Omicron infection. But what surprises us is that when we look at Wuhan spike, the advantage conferred by Omicron infection is still observed, but only for IgA. And so this suggests that any kind of mucosal priming, even with a variant that's very different than the original vaccine, can potentially boost the mucosal immune response. And so we're right now just waiting with bated breath to test other spikes from other non-SARS-CoV-2 coronaviruses, such as SARS-1, to ask if there's any further breadth of this immune response at the mucosa. So so the summary of those findings are that 3 COVID-19 vaccine doses provokes less anti-Omicron spike IgA and IgG in the saliva compared to 2 doses plus a breakthrough, and that these 2 doses plus an Omicron breakthrough actually enhances the anti-Wuhan IgA response, but not the IgG response. And that's presumably due to some kind of local recall. And so in future, what we want to do are really ask, how are IgA plasma cells generated in response to systemic mRNA administration? and compare that with a systemic versus systemic followed by intranasal administration. And how does that compare with infection? Where do these IgA plasma cells reside? We've only had saliva to study in humans. So in mice, we'll be able to look at different tissues and ask whether these plasma cells are sitting in the upper respiratory tract, or do they leave? Do they go to the gut? All sorts of neat things we can do in mice. And again, what about other cerebral coronaviruses? And I'll stop there and hand it over to Kelly, but I think I've acknowledged the students as they go. But in particular, I want to acknowledge funding from CITF and from CIHR and COVARNet. Thank you.
Thanks, Jen, for that fantastic presentation and a real great overview of everything COVID and mucosal immunity related. Just in terms of time, we only really got time for one question, so I'm just going to ask this question that Pascal's put in the chat. And Pascal, if I get it wrong, please feel free to unmute and ask it properly. So I think he's referring to the data that you showed from the Sheba and Toronto cohorts and your comparisons between cases and controls. So do you know any information in terms of the differences between age, sample timing, dose intervals? Is there any matching that was done, and can any of that explain the differences between the IgA levels? And while you're answering that, I'm just going to pop up one slide to just speak to before we hand over to Kelly.
Sure. So, and sorry for going too long, I lost track of time. But Pascal, so the Sheba cohort was case control matched. So there you can check out the New England Journal paper that was first published to show the case control matching algorithm. But there was, yes, so there was matching for age, sex, and then everyone got the vaccine according to the monograph. Oh. So that was controlled for, if that's answering your question. I hope it is.
Yeah, it does. Thank you.
Okay.
Beautiful talk. Thanks for coming all over the BC.
Okay.
Yeah, much easier on Zoom. So I just want to do a brief plug just for upcoming webinars. So our next CIC BC Research Symposium in June, glad to say we have Dr. Peter Brodin, who will be presenting to us on the work that he's been doing in Sweden and in the UK. And then from the CAIRE side, we have a shared symposium on human challenge trials in a couple of weeks with Dr. Chris Chu from Imperial in London and Dr. Scott Halpern in Dalhousie, who've both been setting up different flavors of human challenge trials. So we'll hear more about that. And then Beth will put links again to the chat for people who joined late. So I'm going to stop sharing. And Kelly, if you want to get your slides up, and while I introduce for people who don't know you, so Kelly McNagny is going to talk to us about COVID-19 infection. So Dr. McNagny obtained his PhD in cellular immunology at the University of Alabama and moved to the European Molecular Biology Laboratory in Germany, where he did his postdoc work on transcriptional control of hematopoietic stem cell maturation and cell fate, and then developed his own research lab here at UBC, and he's currently a full professor in medical genetics and interim co-director of the Biomedical Research Center, and has received a number of awards, including a CHR Scholarship, Michael Smith Foundation, Senior Scholarship, and the 2002 Shao Pfizer Junior Faculty Award from the American Association of Immunology. So I'm going to hand over to you, Kelly, and just thank you again for stepping in at the last minute. I know it was Melina Messing, who's one of your students who was supposed to give this talk and couldn't join us today at the last minute. So over to you.
Thanks so much, Manish. Really appreciate it. And yeah, you're getting me as a pinch hitter because Melina couldn't talk today because of an illness, but you'll have to suffer with me giving a second-rate story. So I'll try to make this very quick, but I think like everybody, when SARS hit, we thought, is there anything that we can do to help out? And knowing that we had no funding and not very little time, we jumped in with whatever tools we had. And at that stage, we were making a CITOF panel of about 35 to 38 antibodies to try to study development of asthma in kids and figure out what causes some kids to get allergic and other kids not to. And we thought, why don't we just apply that and a couple other omics techniques to studying patients and see if we can figure out why some people do poorly and why some people do well. And I think you all know that, when you get a SARS infection, you can have anything from completely asymptomatic to mild symptomatic to going to the hospital because you're not really feeling well, to winding up in the ICU, and then a small percentage, 1 to 0.1%, wind up dropping dead. And looking at that continuum, that has a potential to overrun healthcare systems very quickly. And we thought, what can we do to help out? And looking first at this area, the early stages, we thought, there's very little we're going to be able to do there. Everybody's getting infected. We don't know when they get infected because testing was pretty bad early on. We don't know how much virus they got exposed to. So knowing what stage of the continuum they're on and whether they're going to get better or worse is really a challenge there and would need a big cohort. And then likewise on the backside, when you start looking at patients and trying to determine whether they die or survive, so it's a little bit like looking at sepsis.
Is this Martin's mom?
Oops.
Okay, good. Well, I got the right person there.
I think if you look at patients with sepsis, it really becomes a coin toss as to whether they live or die, and trying to discern a signature at that stage is really a challenge. And so what we focused in on was patients who just came into the ICU. And the idea was that, at a certain stage, you get sick enough to be admitted to the ICU. So it's narrowing the boundaries a bit. And it's usually before you've had a lot of therapeutics. So we can try to diagnose a signature that will then tell you whether you're going to recover quickly or spend a long time in the ICU and or die. And we put death in this category too, because we thought, if you die shortly after being admitted to the ICU, that just tells you went there a little bit late, and you should be in with the patients that wound up for a long time in the ICU. So that's what we did, focused on severe disease, day of admission to the ICU, take the blood. And the way we analyzed things is we took healthy controls that were age matched, patients who had been in the ICU for 6 days or less, and patients who wound up being in the ICU for more than 6 days and or died. Took blood from them on the day they were admitted, looked at the serum for cytokine levels, looked at the peripheral blood markers to see what cell types were in their blood, and then tried to predict outcome based on looking at those parameters. So we had 90 patients where we had all the cytokine measurements. We had an initial cohort of 14 patients where we knew some lived and some died. And then we had another cohort of 28 as a replication cohort. And just to show you some of the parameters, again, we set an arbitrary cutoff of 6 days in the ICU and then allowed to go home versus 6 days plus or died. So that's how we separated the patients. Looking at the day of admission, you can see that the ages were similar between the cohorts, body mass index, inflammatory markers in the peripheral bloods like CRP and D dimers for clotting, all fairly similar between the patient cohorts. There were more PMNs in the patients that were in the ICU, and that makes sense because they've got a viral infection, and a few less PBMCs, and that's because we know there's a little bit of lymphopenia that comes with COVID infection. Okay, so starting with the cytokines, we looked at quite a few of them. One thing that really jumped out was if you had elevated levels of IL-10, you tended to be in the long stay or dying cohort rather than the short stay cohort. And that's been seen by Akiko Iwasaki now as well, and a few other groups. High IL-10 seems to be a diagnostic marker for patients who are really having a bad bout of COVID. Same was true with TNF. In fact, it was a little bit better statistically. And again, this is the day of admission, but then if you follow those patients through their time in the ICU, you got an even better statistic. So looks like IL-10 levels really pick out the patients who are going to have a bad outcome with COVID. We can talk about why that might be a little bit later. Looking at the cellular markers, again, we had 38 markers. That was able to break our peripheral blood samples into 44 different clusters. If you just look simply at major clusters of blood cells in these patients, so B cells, CD8 T cells, CD4 T cells, myeloid cells, I'll show you, it really doesn't tell you very much. So here's CD4 T cells. Healthy controls have plenty. You have a bit of a dip in patients in the ICU, but you don't have a signature that can allow you to distinguish the short stay and the long stay cohort. Same with CD8 T cells and B cells and NK cells. Big drop in mate cells, and that was pretty interesting to us if you look. So that distinguishes people who are in the ICU, but it doesn't allow you to separate the two cohorts. And the same is true for plasmacytoid dendritic cells. So really, if you just look at major subsets, it's still very difficult to separate the cohorts. So then we said, well, look, let's do one step further. We know there's an expansion of myeloid cells in the patients in the ICU. Let's really drill down and see if we can find any signature that distinguishes the ones who are going to have a good outcome from bad outcome. So we took the non-T cells, focused on GM-CSF receptor positive cells, and then with that group of cells, we clustered them based on these myeloid markers. If you do that, and it's important to restrict to just a few myeloid markers, because if you put in the whole 38 antibodies, you start getting too much noise. Once you do that, now you start getting really nice signatures. So here's the initial cohort, healthy control, less than 6 days in the ICU, low level of these classical monocytes, but they're a little bit distinct because they have low levels of CD11C expression. That's the one marker that allowed us to separate these patients really well. Long stay had a much higher level of those cells. So that was true in our initial cohort, more robustly in our second cohort, or our confirmation cohort. And if you want to look at how these things look in terms of expression, here they are by flow. So classic monocytes are CD14 positive, LPS receptor positive, and FC receptor negative. Send those to this gate. And this is looking at now CD11C expression. So in the healthy controls in the short stay patients, very few of these cells. And they start creeping up in the patients that are going to either die in the ICU or stay for a really long time. So now let's see how well we can pick out the patients based on those parameters. So again, with IL-10 versus TNF, here's the patients that have above 15 picograms per mil of IL-10. All of these are the patients in the long stay cohort. So that's already a pretty good way to bin patients. Here are a few patients who wound up being long stay or died who are still falling in with that negative cohort. So we got to do a better job of resolving them. If now, instead of just focusing on IL-10 and TNF, you do IL-10 versus these classical monocytes with low levels of CD11C, now you really get good separation. You can pick out with 91% specificity those patients who are going to be in for a long time and 91% sensitivity. In fact, you just have a couple of patients who are leaking through in this assay. So to make a long story short, we focused in on ICU admission as our time for analyzing patients. That's really the biggest burden for the hospitals. If you just simply look at the level of IL-10, 15 picograms per mil, and you look at these classical monocytes that have low levels of CD11C, you can pick out with almost 100% accuracy the patients who will easily come out of the ICU within 6 days without needing any extra care. And those patients that are likely to going to be there for a long time and or die. And that might give you a better way of addressing the patients that really need your urgent care, and might also give you an ability to start monitoring their response to therapeutics. So that's where we'd like to go next. That's the quick talk. Melina Messing is the hero of the story. She's my graduate student, fantastic graduate student, did virtually all the work I showed you here. And then another big shout out to Kevin Leslie, who some of you may know. He's retired at a very young age because of some really nice antibody production work he did with Amgen, and came out of retirement just to help us do some of the statistical and cell type analyses. And I will leave it there.
Thanks, everyone. So I think I will draw it to an end there. So again, thank you to Jen and Kelly and for everyone for joining us today. So thank you again so much, and hopefully we'll see many of you again for one of our future events.